for thank you everyone for joining this Connect seminar series. And before I start introduction of Greg, I want to say that during the talk, or if you have any questions after the talk, please enter them in the Q&A tab, not in the chat. And then I will read it out to Greg and he can answer them. If we don't get to all the questions, Greg will be forwarded the questions and he will respond to you directly. So with that off the agenda, so it is my true honor and great pleasure to introduce colleague, friend, Greg Hannon. So I met Greg when I joined Cold Spring Harbor a long time ago. So Greg is currently the director of the Cancer Research Institute in UK, Cambridge. Before that, he started his faculty position in Cold Spring Harbor back in the 90s, 1996. Became a professor there, and then subsequently moved to, recently moved to Cambridge, UK. And he is now directing a lab that relates to what he's going to talk about, including RNA and vascular mimicry. So Greg is a highly accomplished scientist. So he received the ASER Outstanding Achievement Award in 2005. He's a member of National Academy of Sciences, an Academy of Medical Sciences, a member of EMBO. He's also a fellow of the Royal Society. And more recently, he became the AACR Research Academy member in 2020. And Greg has, I think he also in UK, he also has this coveted Fellow of Trinity College, a position there that is very hard to come by, very hard to get into. He has served on numerous boards and committees. He has published over 300 publications, founded 6 companies. So the list is very long and I don't want to take Greg's time. And some of his major discoveries related to discovery of tumor suppressor genes when he was with David Beach in Cold Spring Harbor, and then subsequently the discovery of siRNA related biology. Today, he's going to talk about something interesting and new, which is vascular mimicry as a mechanism of resistance to anti-angiogenics. So with that, Greg, take it away.
Thanks very much, Senthil. Sorry we're running a little bit late. We had some snafus probably on my side getting onto the Zoom. It's a pleasure to do this. I wish I could be doing it there in person. We had the great pleasure of having Senthil over a couple of weeks ago. And I have to say, the feedback both on Senthil's talk and on just having in-person interactions scientifically again was just amazing. So I find it distracting to have a camera on while I'm talking to my slides. So I'm going to turn my camera off and just focus on the presentation, but I'll come back on camera when we get to the questions. So with that, here we go. So the work I'm going to talk about actually started about 10 years ago when we became interested in tumor heterogeneity and particularly in its consequences for the course of disease and response to therapy. And I think I don't need to go through the concepts here. I think we're all quite well aware that tumors are heterogeneous, both between patients and within a patient's tumor. They're composed of both genetically and phenotypically distinct clones, with each with their individual propensities and properties. Now, we at the time were struck by the fact that most studies of tumor heterogeneity were retrospective. And that's still true today, where the heterogeneity is cataloged, for example, at the single-cell RNA-seq level or at the level of single-cell genomics. And I just put up a very early kind of inspirational paper from an ex-colleague of mine at Cold Spring Harbor, Mike Wigler. Now, as I said, most studies are retrospective, but that's changing. On the right is a new study led by Saurabh Shah and Sam Aparicio, part of our RCR UK Grand Challenge team, where they use single-cell genome sequencing to track clonal fitness under stress. In this case, the stress is therapy. So it is changing, but it's, I think, changing slowly. So our focus was not on cataloging tumor heterogeneity, but instead understanding how phenotypically distinct lineages contribute to disease. We wanted to know not just that the heterogeneity was there, but we wanted to address precisely what the individual capacities of each of those lineages were. So we set up a model system, which we still use today, but it was based on the idea of taking a model cell line, 4T1, which is a mouse model of basal, claudin low breast cancer, that can be transplanted syngenetically into mice. So you have the benefit of doing the experiments in the presence of an intact immune system. We introduced into this population of 4T1 cells a set of viral DNA barcodes so that we could individuate lineages. And then we separated those lineages by single cell sorting and then growing out individual clones, which we then mixed back together in equal numbers as our kind of simplified model of heterogeneity. Then we tend to transplant those orthotopically into the mammary fat pad and then do a number of different things to the mice. In the case of what I'm going to talk about today, it was really harvesting the primary tumor and then harvesting cells at secondary sites and comparing the clonal contributions to each of those. Now, in this model, we set out, student and postdoc, Elvin Wagenblast and Simon Knott, to generate a heterogeneity model that I thought would have, hundreds of components. In the end, when all was said and done, we had 23. But as we found over the years, that's actually enough to capture a lot of phenotypic heterogeneity, particularly in this quite genomically unstable model. So, as I said, what we did was we harvested the primary tumor, and then secondary sites. We took lymph nodes, brain, liver, lung, and collected circulating tumor cells from blood, sequenced the DNA barcodes, and this is just a heat map of clonal prevalence in each of those samples. Now, the primary tumor, which is not well represented here, I think that's probably cut off from a larger figure, had at early time points a pretty equal mixture of all the clones. At later time points, it came to be dominated by a few clones, including clone L. But the clones that dominated in the primary tumor, the lineages that dominated, were very different than those that we found at secondary sites. And in fact, we found two clones, which we called E and T, which were very good at being present in the blood, even though they were a minority species in the primary tumor. And it's clone T that had the greatest metastatic competency, but that's probably a story for another day. And so we became interested in why these two clones, E and T, were so successful at leaving the primary site and existing in the vasculature. And I don't have time to go through it all, but to make a very long story short, we believe, and our data has supported, that these clones are able to create structures in the primary tumor known as vascular mimics. Now, vascular mimicry was discovered in 1999 by Mary Hendricks in melanoma, and it has been a contentious field. And so, we have been working, we continue to work, and I'll come back to some of this in a minute, to really understand what these structures are and what their significance is. Now, of course, we all know that tumors have to recruit a blood supply to deliver oxygen and nutrients. The conventional way that they do this is by promoting angiogenesis. And it's really this that led Judah Folkman so many years ago to propose that, targeting this process in the less plastic, the less genetically unstable, and less malleable components of the tumor would be a successful anti-cancer strategy. And that in turn has led to the development of anti-angiogenic therapies, and we'll come back to that later. But I think there's a growing appreciation, though, as I said, still controversial, that there's more than one way to skin a cat, and there are alternative ways that tumors can adopt to allow their perfusion. And one of those alternative ways, as I said, is this phenomenon of vascular mimicry. And what happens in vascular mimicry is tumor cells form channel-like structures lined with ECM. And those channel-like structures are thought to hook up to the normal blood vessels in proximity to the tumor and allow blood flow to allow oxygenation and nutrient delivery. This is increasingly appreciated, or the presence of these structures is increasingly appreciated as being associated with poor prognosis across many different tumor types. And in fact, the way you identify these structures histologically is by looking at the staining of that ECM-rich basement membrane and seeing it occur in the absence of staining for an endothelial cell marker, generally CD31. Now, this is an example from small cell lung cancer from Caroline Dive's lab. Sorry, the image on the left is a histological section taken from a circulating tumor-derived xenograft showing the presence of both normal endothelial line vasculature and these strange sort of channel-like structures. And in many of the histological images that she has from these CDX tumors, you can actually see red blood vessels in the vessel-like channels that are lined by tumor cells. On the right is a complementary study that she did in patients, looking at histological analysis of samples, again, from small cell lung cancer, and quantifying the ratio of these CD31 negative channels to CD31 positive vessels, and showing that the presence of relatively high levels of these vascular mimics is associated with poor outcome. Now, if we apply those same techniques to measure the prevalence of these channels in our 4T1 model, what we see, and this is an assay which we've done where we created monoclonal tumors from each of the 23 components of the heterogeneity model. And if we simply count CD31 negative past positive channels, again, we see that clone E and T are the most proficient clones at actually creating these channels. There is an in vitro proxy assay for this, for being able to make these structures in vivo that involves the creation of tubes when you plate cells in a 3D matrix, in this case, Matrigel. And if you look at the panels on the right, what you can see is that the parental 4T1 line, and I actually don't know if you can see my cursor, but the parental 4T1 line or this 4T1L clone that dominates the primary tumor at late time points don't make these structures, but E and T do. And these are just two example human breast cancer cell lines that also form these structures in Matrigel. And again, I'm not claiming that these are vessels, but it's a proxy assay that correlates with the ability of cells to create these channels in tumors in vivo. So this indicates that we have two VM proficient subclones in 4T1, 4T1E and 4T1T. But of course, the question is, if we look in vivo at these structures that are prevalent in these particular clonal tumors, are they relevant? Are they actually perfused? And so we've been working on this for quite some time, and we've looked at this in a number of different ways. We've looked at this by serial 2-photon tomography. We've looked at this in this case by light sheet, and we've just gotten started with a collaboration to do serial block-based EM, but I don't have any results from that yet. But this is an experiment where we took a 4T1T tumor and we perfused the mouse with a fluorescently labeled lectin just before sacrifice. We then stained this sample with anti-CD31 and cleared it and imaged it on the light sheet. And so this is sort of the high-level view of the sample. But if you zoom in, what you can see is, again, an abundance of these CD31 lectin positive, double positive structures. But you also see these channels that are negative for CD31, and you see them, doesn't matter whether you slice it X, Y, or Z, in exactly the same plane as the CD31 positive channels. So I don't think this is an issue of antibody penetration, but they're clearly labeled by the lectin that was introduced into the circulatory system. So this provides at least some evidence, and I wouldn't say that it's definitive yet, that these structures are somehow connected to the normal vasculature, and they can receive fluid from it. And if the size of these is easily comparable to even the slightly larger CD31 positive vessels that we see, and certainly large enough for the passage of red blood cells and immune cells. So the question that we had was, how do these cells acquire the ability to create these structures in vivo? And what are the characteristics of the cells that endow them with this property? And so, since transcription factors often act as master controllers of cell fate and cell state, we started off by looking for transcription factors which were differentially expressed in clones E and T, the VM proficient clones, versus the remainder of the tumor. And if we did this, FOXC2 stood out as the most significant differentially expressed transcription factor. And this was interesting because FOXC2 is well established as a key transcription factor in embryonic vascular development. And it's also known to regulate some endothelial cell gene expression patterns. So FOXC2 looked like an interesting candidate for driving VM proficiency. So the question was whether FOXC2 impacted the ability of cells to create these structures. So to address that question, we identified a couple of effective shRNAs that can target FOXC2 either in mouse or humans. We did the knockdowns, and then we did initially this proxy assay which is tube formation in vitro. And so again, here's the control shRNA, and either of the two FOXC1 targeting shRNAs negated the ability of these cells to organize into these structures. And if you quantify this, it's quite a significant change and quite a significant loss of the ability to create these structures in matrigel. But of course, the question was what happened in vivo. And so we took these knockdown cells, we injected them orthotopically into recipient mice, and quantified CD31 positive vessels and CD31 negative channels. And what we see when we compare those ratios is that either of the two knockdowns of FOXC2 shRNAs 4 and 6 reduce the prevalence of the CD31 negative channels. In fact, if we quantify that, we not only see that the CD31 negative channels are greatly reduced, but we see an increase in CD31 positive vessels. Okay, so the question is, what does FOXC2 do to promote this? Now, FOXC2 has previously been annotated as a driver of the epithelial to mesenchymal transition. And so we wanted to know whether there was an involvement of EMT in VM. And so we went and basically started mining published gene expression data and generating some gene expression data of our own. This is some published data where FOXC2, or these canonical sort of EMT drivers, were overexpressed in vitro transformed human mammary epithelial cells. And what we see is interesting, which is that FOXC2 drives the program. And this is tissue TSEA, so tissue set enrichment analysis. And what we see is that FOXC2 drives a gene expression pattern where the tissue is representative of blood vessel. The question is, and that doesn't happen if you overexpress twist or snail. And so the question is, what components might be underlying this expression signature. So we did gene set enrichment analysis. And what we see is that FOXC2 overexpression enriches gene sets that are defined as endothelial enriched genes, and knockdown reduces the expression of these. And so what we think is happening is that FOXC2 isn't driving a conventional EMT, but is promoting the expression of endothelial signatures in a sort of EET, or an epithelial to endothelial transition. And that doesn't mean that there isn't involvement of an EMT, and I'll come back to that later. So we're seeing FOXC2 drive expression patterns that are enriched for endothelial genes. There's another assay that people use for isolation, in this case of endothelial cells, which is that they have the property that they will take up acetylated low density lipoproteins. And so we asked whether our VM proficient cells would take up ACLDL, and whether that was impacted by FOXC2 expression status. And so what we see is that, so if we look at 4T1L, right, so that's the clone that is not VM competent, dominates the primary tumor at late time points. It will not take up acetylated LDL. 4T1T, VM competent, takes up acetylated LDL, but it loses this ability if we knock down FOXC2. And we can also take a variety of different human cell lines that we've also shown can make these structures in vitro and in vivo. They all do take up acetylated LDL, or a subset of cells in the population does. But again, you either reduce or negate the ability of these cells to uptake acetylated LDL if you knock down FOXC2. So it looks like FOXC2 is driving these cells to have at least some components of endothelial expression signatures, and that translates into at least one property that is prominent in endothelial cells. So the question was, where do we find these? Where do we find these characteristics? Where do we find these properties? And so, focusing initially on breast cancer, we went and looked at the breast cancer cell lines in the CCLE. We looked at a large collection of patient-derived tumor xenografts from Carlos Caldas, and we looked at core FOXC2 target genes and where they were enriched. And we look at FOXC2 targets more than FOXC2, because FOXC2 is also regulated at the level of protein localization. So FOXC2 can be expressed, but in some cases it's retained in the cytoplasm. And then we looked also at endothelial genes, again, refined from a GSEA list, where we've removed some genes that are also shared with mesenchymal cells. And what we see is that FOXC2 core target genes show higher expression in basal, claudin low breast cancer cell lines. And again, this is the type of breast cancer that 4T1 is supposed to best model. We see the same thing if we look at the human-derived reads from the PDX models, but we don't see any difference if we look at the mouse-derived reads. And we see very similar pattern for the endothelial expression signatures that we think FOXC2 helps to drive either directly or indirectly. Now, this is just looking at 71 different PDX models from the Caldas lab. And the idea here was to try to predict, based on the expression of FOXC2 targets, which of these might be able to make these vessel-like channels in vivo and which ones would not. And so we've looked at a number of these, and the correlation is very good. So I'll just show you two examples. One is, I think, this red dot, and that's HCIO10. And then the other one is this blue dot, which is AB551. And again, quantifying the ratio of PAS positive CD31 negative to pass positive CD31 positive, there's really an enormous difference between these models. And in fact, if you look here, you're hard pressed to find a CD31 positive vessel, even though by all measurements, these tumors are not hypoxic. So then the question was, if you have an alternative mechanism for perfusing the vasculature, or for perfusing a tumor that does not rely on conventional pro-angiogenic signals, the question is, then does VM, this ability to make these kinds of channels, influence responses of tumors to anti-angiogenic therapies? And this has been hypothesized for a long time. But strong evidence supporting that hypothesis has not been forthcoming, in part because knowledge of ways to predict VM propensity and knowledge of ways to experimentally manipulate VM capacity have not existed until fairly recently. So we could start by asking whether the gene sets that we've identified, these core FOXC2 target genes, have any correlation with resistance to anti-angiogenic therapy. And here we've looked at a couple of different experimental cohorts. These are published data sets. This is a gene expression data set from a clinical trial conducted in breast cancer patients by Adrian Harris at Oxford. This is a GBM xenograft in vitro or animal model trial. And in both cases, we see the genes that are differentially expressed in those patients or models that are resistant to anti-angiogenic therapy actually are strongly enriched for FOXC2 core target genes. Oops, sorry. And you see exactly the same thing in GBM. And actually, if you look at the genes that are differentially expressed in the components of our models that are competent for ACLDL uptake, then you see, again, an enrichment for the differentially expressed genes that are characteristic of those patients, in this case, that are resistant to anti-angiogenic therapy. So that's, and we've done this across lots of clinical data sets that are available in the literature. But of course, the key for us is that we had models where we knew VM proficiency and where we could manipulate it. So could we ask in an experimental model whether the ability to execute this process had any relationship to sensitivity to anti-angiogenics? Okay, so on the left, we're comparing the sensitivity to B20, which is kind of the mouse version of bevacizumab, and its impact on either parental 4T1 cells, which are largely VM incompetent, though they must have some small component of VM competent cells because that's where the Ts were derived from, for the highly competent 4T1T lineage. And what we see is a drastic difference in the response of these tumors to anti-angiogenic therapies, with the 4T1T model responding virtually not at all. And we see a similar, although perhaps slightly less dramatic, pattern if we treat these same animals with axitinib. So it looked like there was at least a correlation between models that can create these channels and models that can't, and their sensitivity to this intervention. But of course, the question was, if we intervened genetically and negated the ability of these VM competent models to create these structures, did we have any impact? And so I could show you this a bunch of different ways. I think the time course is one of the easiest ones. So basically what we did was we took 4T1T and made knockdowns using either of two shRNAs or the control shRNA, orthotopically transplanted these into mice, let the tumors form, and then treat either with or without the B20 antibody. And what we can see is a highly statistically significant increase in the sensitivity of these otherwise resistant models if we knock down FOXC2. So these are the two FOXC2 knockdowns, and the corresponding colors are minus and plus treatment. And the and the black minus and plus treatment is the control hairpin. So it remains highly resistant to this intervention. So it looks like if we manipulate the ability to create these structures, or alternatively, the underlying characteristics of the cells that can create those structures, we modify sensitivity to anti-angiogenic therapies. Now, of course, anti-angiogenic therapies create changes in the tumor microenvironment, hypoxia, pH changes, etc. And so, the question is, did that have any impact on the ability of cells to execute this developmental program? And so the idea really was, if we have a very small percentage of, let's say receptive cells, right? Perhaps they have higher FOXC2 expression, perhaps they have additional characteristics that I'll talk about in a minute. If they're exposed to a hypoxic environment, does that provide a selective pressure? Does that provide signals that might enhance the proficiency of these cells to create these structures? So what we did is we took our parental model, and we simply grew tumors, again, orthotopically, treated with anti-angiogenic therapy, and quantified the presence of CD31 positive vessels versus CD31 negative channels. And and what we found was that actually, if we exerted selective pressure in this parental, which is poorly VM competent, exerted selective pressure by treatment with anti-angiogenics, we see across many animals, you form fewer CD31 positive vessels, but you form many more of these past positive CD31 negative channels. So the question was, is this selective pressure? Is it signaling? Is it a combination of both? So we asked the question of whether FOXC2 expression levels had any impact on the ability of cells to exist in a hypoxic environment. And was there any relationship between gene expression patterns that are characteristic of hypoxia and FOXC2? And I probably would have said anything about this if the answer wasn't yes to both of those. We can take parental 4T1 cells, expose them to quite severe hypoxia. And in that case, knockdown of FOXC2 compromises their ability to survive. And if we take a different model, again, also competent to make these structures, the MDA and B231 cells, and we compare genes that change in their expression status when we knock down FOXC2 versus control knockdown. And this is just gene set enrichment analysis across many, many, many gene sets. The ones in red are those gene sets that are annotated as being associated with hypoxia and hypoxia responses. So it looks like there is a relationship between FOXC2 survival in hypoxia and expression of hypoxia related genes. So the question is, if we took cells and we exposed them to hypoxia, did that have any impact on the ability of the cells that had been exposed to this condition to form these CD31 negative channel-like structures in vitro and in vivo. And that indeed was the case. So here, these cells have been cultured at atmospheric oxygen. These are the cells that were selected for survival under severe hypoxia. And this is, remember, from parental 41s. And we see a very large increase in the ability, again, in vitro proxy assay, but correlates with the creation of these structures in vivo. We see an increase in the ability, either because of signaling or selection. And at this point, we actually don't, strictly speaking, know which one that is. However, we have seen that exposure to hypoxia, in this case, 1%, not 0.1% oxygen, does cause an increase in the expression of FOXY2 target genes. And in fact, if we treat with this HIF1, HIF2 inhibitor, which again, taken with a grain of salt because there have been some questions about its specificity, that partially negates the increase in expression of these FOXY2 target genes. And we see the same if we look at these sort of core endothelial gene expression, endothelial gene sets. And so hypoxia does seem to have an effect on FOXY2 targets, and also in turn on these gene sets that might be a marker of this epithelial to endothelial transition. So sort of finally, we became interested in, what is it that makes these cells susceptible to this change in cell fate or cell state that's driven by FOXY2. Because we know that not every cell within even the clonal lineage represented by the VM competent clone T can participate in making these structures. So we thought, there must be additional characteristics, and there's probably several additional characteristics. And so we wanted to know, not by looking at gene expression patterns in the end state, once these kinds of structures had formed in vivo, but by looking at cells that are able, under the right conditions, to undergo this transition. What were their properties? And so we took advantage of an approach that we had developed recently, which lets us simultaneously follow lineage dynamics, single cell gene expression patterns, and we think also single cell whole genome sequencing all at the same time. And it's a fairly straightforward approach. We've got a neutral barcode that's integrated into a lentivirus. And sort of much like we did in the very original experiment that started us down this path. But in this case, that barcode is readable in single cell RNA-seq. And so basically, what Sophia and Kirsty have been working on is engineering cell populations using this, this lentiviral barcode library and a procedure we call wild-seq, which is not a coincidence considering the PhD student who developed it. And then sort of very importantly, and a skip that is completely glossed over here, it's exceptionally important how you bottleneck the populations, because we have to hit a sort of clonal representation where transplantation into replicate animals really does allow you to visualize the same subset of clones, so that you can actually gather meaningful comparisons. So an awful lot of work has gone into that. And then that bottlenecked population gets transplanted, again, multiple replicate animals. The animals can be exposed to a variety of different selective pressure stress conditions, in this case, anti-angiogenic therapy. You then recover the tumors from the treated and untreated animals and do single cell RNA-seq and look for patterns that associate with particular responses. And so in this case, what we're looking at is a set of clones engineered with these vectors. And you've got a few clones that are reasonably highly represented in the tumors of multiple animals that basically don't change in their abundance upon treatment with an anti-angiogenic, in this case, axitinib. You have a few clones, a couple here, that actually become dominant in the tumor when treated with AAT. And in fact, when these clones become dominant in the tumor, they also see an increase in the percentage of cells which are representative of these clones that increase their expression of these endothelial gene sets that I discussed earlier. So in this case, we've got these two clones, and we asked, well then, what are the properties of these two clones that seem to correlate with the ability to do this, right? So, and that's ignoring FOXC2 for the moment. And what we find is that clones 662 and 673 are highly enriched for cells that show both epithelial and mesenchymal characteristics at the same time, these so-called hybrid epithelial mesenchymal state cells. And I'm just showing you here the percentages scored by co-expression of fibronectin and e-cadherin, but the same thing, but you get the same result if you use other epithelial and mesenchymal markers or combinations thereof. And so what we think we're seeing here is that you've got cells that are in a highly plastic state, somewhere along the continuum between epithelial and mesenchymal. They're receptive to the signals provided by FOXC2. And we think it's really these cells that are the ones that both show resistance to anti-angiogenic therapies and are able, in a correlated way, to form these CD31 negative pass positive channels in vivo in tumors. So just to kind of finish up there with some take-home messages. So what I hope I've shown you is that tumor cells can form discrete ECM-rich tubes that can be labeled by IV lectin injection and are therefore at least in some way perfused. What I haven't shown you is many H&E images where we see red blood cells in these channels and STPT images where we can see at single cell resolution groups of tumor cells surrounding a hollow lumen. What we haven't yet identified, and it's possibly because we just haven't had time to delve deeply enough into the light sheet data, is a picture where there's definitive connection between these structures and the endogenous vessels, and that's something that we're working very hard to do. So I've shown you that proficiency for forming these structures is driven by a pioneer transcription factor, FOXC2, which is a transcription factor important for embryonic vascular formation. That FOXC2 target genes correlate with the capacity to do this process, vascular mimicry, across multiple solid tumor types, tissue types, models, and that FOXC2 target genes correlate with poor response to anti-angiogenic therapies, both in models and in human patients. We can experimentally manipulate FOXC2 expression that influences response to AAT. And there's the possibility that the hypoxia created by treatment with anti-angiogenic therapy may in fact activate the capacity to do vascular mimicry, sort of creating a vicious cycle where the treatment sows the seeds of its own resistance. And the prevalence of a hybrid EM state correlates with VM potential. And this is interesting for lots of different reasons, not the least of which, that Senthil and I spent many happy hours over beers talking about plasticity being the root of all evil in cancer about 20 some years ago. But also because it may be relevant in terms of how these structures form, perhaps by communal migration of clonal lineages that carry these properties. And in fact, what I haven't shown you is a lot of 3D imaging by serial 2-photon tomography over the time course of development of these 4T1 tumors. If we introduce or create a tumor that has 20% fluorescently labeled T clone and then a background of either L or the parental, we see the T clone moving through the tumor in well, I can't say moving, it's organized within the tumor in what looks like streams of cells. And so it's possible that, it is a consequence of that communal migration that these kinds of structures form. And we're really interested in trying to watch that now by intravital microscopy. So I'll stop there and take questions. I want to thank the people who were really responsible for doing the work that I presented. So for a while, when I was in transition, I left New York for Cambridge about 7 years ago. But for a while, I had a lab at the New York Genome Center, and it was pretty much run by this guy, Ian Cannell, who is responsible for a great deal of the work that you've seen today. Isabella Pearsall was an RA with Ian at the Genome Center and is now a PhD student in my lab in Cambridge. In my lab and in the CRUK Grand Challenge team, I talked about work from Sophia and Kirsty, who are really responsible for a lot of the lineage mapping and single cell RNA sequencing, and actually tons of single cell RNA sequencing that I don't have time to show you. And Julie Alerta and Carlos Caldas have been instrumental in our analysis of the patient-derived xenograft models. And in fact, just as a last anecdote, it's an interesting story that Carlos had very little interest in this work for a while until he gave his PDX models to a colleague at the Weitzman, who rang Carlos back and said, I don't understand this. Your models don't have any mouse endothelial cells in them, but they're not hypoxic. They're perfectly happy. And that was really the trigger that got Carlos working on this with us to try to understand what he was seeing in the PDX models. So with that, I'll thank you all for your attention. Zoom is a very strange way to give seminars because you have no idea what faces your audience is making at you while you do it. But I'll stop there and then take questions and turn my camera back on and see if we can't get this to work. Right.
That was fabulous, Greg. I will clap on behalf of all the audience. There are a few questions in the Q&A, but before I get to it, I want to ask you a couple of things. Maybe
Sure.
One thing. I'm curious about whether you have thought about whether the tumor cell vascular mimicry based ducts actually make a direct connection to the main endothelial lining or that come close to the tumor, or are there vessels coming from the tumor that are coming from the vessels that are endothelial cells into the tumor, and the vascular mimicry is an intra-tumoral phenomenon, meaning it never actually makes that plumbing connection by itself directly to the vessel, but actually makes connections to the microvessels that are inside the tumor.
Yeah, I don't think we know, right? And I think that's one of the things that we spend a lot of time looking for, and now that we have the ability to dial this up and down, I think it'll be much easier. But I think we have to bring a whole load of imaging methods to bear to understand this. So everything from high resolution photoacoustic imaging, where we can see whether these structures are carrying oxygenated hemoglobin, to all the way to sterile block face EM. But in between the light sheet and the STPT data, I think is probably where we're going to get the sweet spot for that. So in the STPT data, we can create very large subcellular resolution 3D images. And the challenge there, quite frankly, has been the vascular staining. So it's really only quite recently that we've gotten good lectin staining. Unfortunately, that particular lectin didn't have a good 2-photon profile. So we now have a different one that's made that has a good 2-photon fluorophore. And so we're going to be putting those tumors on the instrument pretty soon. But I think, that is the question, right? I mean, we have seen some crazy things, vessels which seem to have, a break in CD31 expression, but the tumor cells are there. Then there's a little segment which is CD31 positive, and but we need to we need to dig and see how the network is plumbed, as you say.
Interesting. I have a couple more, but I will wait. I'll ask a few questions from the audience, and then I'll come back to this. So one
Yep.
is from Every Mersick. What are your thoughts on the basic origins of the VM? Is it about plasticity-mediated response to hypoxia that activate a differentiation program? Can normal breast, within brackets, stem cells, differentiate into endothelia, or is this a tumor-specific phenomenon?
Well, yeah. Let me preface by saying I don't think there are any tumor-specific phenomena. I think tumors hijack other biology. I'm sure someone will cite an instance, but I tend to approach this by thinking that they don't invent their own. We've been so whether I think normal breast stem cells, whether they can do this or not, I don't know. I'm not sure under what circumstance they would need to. But when we think of a normal correlate of this, we're much more thinking about sort of trophoblast infiltration of of the placenta, and it's a process that superficially shares some of the structures and properties that we see in these channels. That to me is a hypothesis. If you want to look back and say, what is the normal correlate of this? Where is it born in a way? That's kind of our working model. We haven't done any work on it yet.
Okay, great. The next is from Hugo Cavallaro. Great talk. Couple of questions. Do angiogenic growth factors, VEGF or FGF2, promote vascular mimicry? Second question is, do VM proficient tumor cells express VEGFR? If not, could this explain their resistance to B20 treatment?
Yeah. So we don't see angiogenic growth factors being required for this in this model, right? I think there are other models where people have seen an impact of sort of VEGF receptor type pathways, but we don't see VEGFR expression in our VM proficient tumor cells.
Okay. The next is from Patrick Anderson.
Yeah.
It looks like you have used CD31 as your standard endothelial marker, as CD31 is known not to stain all types of vessels. Have you looked at additional markers, example CD34, VWF, VEGFR2, CD105, to confirm that you're looking at structures lacking endothelial cells?
Yeah. So I think this is a totally fair question. We've looked at CD34, and we're still working on getting CD34. We've mostly looked at in the imaging mass cytometry, and we still don't have antibodies to the other three that work in IMC.
Okay. The next is from Rakesh Jain. I think it looks like a comment.
Yeah, that's fine.
But
from Rakesh, I will take the first three words of that comment quickly.
Really elegant work. Your work provides additional support for improving vascular perfusion and oxygen levels in tumors by using vascular normalizing doses of AAT rather than starving tumors. In fact, all AAT trials increased perfusion, oxygenation, correlated with improved patient survival.
Yeah. I definitely appreciate the comment, and I agree that if you went to vascular normalizing doses, you likely wouldn't be driving these kinds of phenomena. And it's not, it probably isn't just all about perfusion. It's also about driving plasticity and motility, and basically creating signaling that pushes you down that sort of both as survival transition to all sorts of therapies, but also potentially increasing the seeding of disseminated tumor cells, which is what we saw in the original paper.
Right. So the next is from Lee Ellis.
Yeah.
Is there an epigenetic cue that induces this mimicry in epithelial cells? Is it more related to a genetic defect in the cells, Or is it possible that epithelial cells taking up extracellular vesicles from endothelial cells?
We don't know, right? We did whole exome sequencing on the clones. There was no smoking gun, but then again, these cells show, more copy number instability than, than they do SNVs. That doesn't mean that, we don't have a mutation in some sort of, regulatory, some sort of, regulatory element or something. So we don't know. I think of this as phenotypic plasticity, because even though it's stable, and we showed that these phenotypes are stable over many generations, 40 population doublings. But we also know that even though that cell was clonal in the beginning, there is a distribution of phenotype. So if it's really genetically hardwired, like if it was a mutation that was entirely responsible for this, I would expect more uniformity.
Yeah.
But, so I think there probably is, whatever you want to call epigenetic, whether it's methylation, whether it's stable gene expression patterns, I think there's an element of that. But there could also be a contribution of mutations. I think I would be, wildly speculating if I were to say anything about taking up EVs from endothelial cells.
Okay. The next is from Michela Corsini. Is there evidence for FOXC2 expression in melanoma PDX or samples derived from mice?
I don't know the answer to that question. If you want to know, email me, because I'm sure Ian Cannell, who lives and breathes this every instant of his life, will know the answer to that.
Okay, you got that. Next, again, is a question from Ugo Cavallaro. Do VM-derived vessels show signs of lower or higher permeability as compared to bona fide tumor vessels? Do they have a parasite coverage?
So they don't have parasite coverage, at least in any of the models we've looked at. And so there again, we're looking mostly by imaging mass cytometry. We haven't done the measurements of leakiness directly, except in the very original paper where we did show that the vasculature in general in these tumors was leaky. But we don't know whether that's, immature vasculature or whether it's the VM vessels. And we would need to go back and start to look at that, so that now that we have much more control over the system, and now that we have imaging methods, where we can really distinctly see these two types of structures. But it is a very good question.
I'll ask the last question. So you showed data for hypoxia-induced FOXC2 and regulation of endothelial cells. And the last bit where you had this EM hybrid state, do you know if that is triggered by hypoxia? Is it also regulated by it? Or is it an independent state that pre-exists in those cells?
We know that it's an independent state that pre-exists in those cells because the comparison that we did was, clone, I think 622, whatever, you get these, we have so many different models with clone numbers, but, what we did is we compared 622 versus the others in the untreated animals. Mm-hmm. And that's where we see a high prevalence of hybrid EM states, right? So that pre-exists in that clone. Whether or not it's also increased by some sort of hypoxia signaling, we don't know. But those clones, those lineages are highly enriched for EM hybrid states, and they are the ones that then respond by upregulating endothelial expression signatures, and they are the ones that come to dominate the tumor post-therapy.
Okay, that'll be interesting. Yes, the hybrid states. So with that, Greg, I want to thank you very much for taking time. I know you got to juggle a lot given the gas crisis and the whole 9 yards.
Yeah.
So I want to.
But we didn't get invaded by our 2.5-year-old, so that was good.
That's good. That's good. It would have been entertaining, though, I would say. It was a true honor to host you, and also it was a great pleasure to listen to this cool data that you have. And hopefully we'll have you here in person sometime soon.
Yeah, I'd love it.
Thank you again for taking time.
All right. And thank you to everyone for listening.